Hello, good afternoon, and welcome to our penultimate seminar of the 2021 seminar series. I'm excited today to introduce our speaker, Dr. Don Yee. Don and his lab group studied the community apologies while I struggle with the mute button. So Don and his lab group studied the community population and urban ecology of aquatic insects, most notably mosquitoes. Don received his bachelor's from the University of Michigan, followed by a master's at Texas Tech and a PhD just up the road from Tyson at Illinois State University. After a postdoc at the University of Calgary, Don joined the faculty at the University of Southern Mississippi, where he is now a full professor. Don is an amazingly prolific scientist with over 50 peer-reviewed publications, and he's getting close to the $2 million mark in grant funding received. Don's work has been really influential to my own, and probably every one of my papers cites at least one of his. Don and I are also what I consider to be scientific siblings, as in we share the same PhD advisor, but we didn't overlap in the lab, and I definitely consider him to be the older, if not wiser, sibling. So today I'm really excited that he agreed to come and talk to us today about some of the work that he's been doing over the last couple of years, working in Puerto Rico after Hurricane Maria. And he actually is one of those scientists that successfully crowdfunded money to get this project going, and in fact, talked me into submitting some of my own money to his crowdfunding. So kudos to Don. So please join me in welcoming Don, and make sure to leave your questions in the YouTube chat.
All right, well, thank you for that introduction. Hopefully I can live up to that short-term and long-term. And I'd like to say just before I start that although Katie's right that we have crowdfunded projects in Puerto Rico, I'm not actually going to talk about any of that stuff today. I'm going to talk about other stuff where agencies have given us some money to do some things. So today is going to be a very broad collection of short stories on work that we have done over the past 4 or 5 years in Puerto Rico, mostly focusing in on disturbance as a factor affecting insect communities. Before I get started, I want to go ahead and acknowledge the students and funding agencies. So the mosquito work I'm going to talk about today was done primarily by 2 of my graduate students, Lee Marie Reyes, who is a PhD student in my lab, Nicole Scavo, recently graduated master's student. And then the other work that I'll talk about today, which focuses on litter invertebrate communities and some other insects, was done primarily by Joe Nelson, Nicole Scavo again, who are both former master's students in my lab. Jacqueline Everly, who's an undergraduate, who is now soon, hopefully, to be Katie and I's academic sibling as she's doing her PhD with Steve Giuliano. Kelvin Reyes and Tesai Mendez, who are undergraduates. And then most recently, Patrick Gilkey, Nate Dahlberg, and Caroline Sori, who worked in Puerto Rico a couple of years ago and are still in my lab. And then funding sources, support from Lakio LTR, which I'll talk a lot about towards the end of the talk, NSF, and then logistical support and other support from the CDC and the US EPA. So with that, I'm going to start and talk a little bit about disturbance. As ecologists, we know that there are a variety of factors that are important in structuring communities. Disturbance is thought to be one of the main ones that exists and is often studied not only across spatial scales, but focused in on particular organisms. So a good definition of disturbance is by Krebs, who says that a disturbance is a discrete event in time that disrupts community structure and changes available resources, substrate availability, or the physical environment. So all of these, this definition encompasses situations in which a community of organisms is affected because something about the environment has changed. It could be the nutrients, it could be the environment itself, the physical environment, and other factors. And there's lots of examples of these. There are many non-anthropogenic natural examples of these, things like large fires, large storms like hurricanes, cyclones, typhoons. but they can also be anthropogenic. They can be man-made. So things like clear cutting of forests can be a disturbance. And at smaller spatial scales, even a footprint can be considered a disturbance depending on the organisms of investigation. So my theme of my talk today is really going to encompass this idea of disturbance as a force that is affecting communities. And since all the work is focused in on Puerto Rico, I want to just briefly talk to you a little bit about the island if you don't know a lot about it. So Puerto Rico is a U.S. territory home to about 3+ million inhabitants. It is located in the Caribbean, about 1,000 miles southeast of Key West, Florida. It's one of more than 30 Caribbean islands and is situated in a place that often experiences rather large disturbances via hurricanes. And these hurricanes are thought to structure the communities that live on these particular islands and also affect their diversity by moving organisms around. So in September of 2017, a rather large hurricane named Maria hit the island. Right before it hit the island, it was a Category 5, which is the largest and the highest category there is, but it was a Category 4 by the time it actually struck Puerto Rico, which you can see here in the center, and it had winds in excess of 150 miles an hour. This is considered the 10th most powerful Atlantic hurricane on record. Now, the records only go back 30 or 40 years, so there may be historically higher ones, but this is considered one of the largest and most devastating hurricane to hit the Caribbean in recorded memory. For the island itself, it caused at least $90 billion in damage. Structures were devastated. The infrastructure was devastated. It took more than a year for electricity to be restored across the entire island. Many roads were damaged, etc. Beyond these sort of human effects, though, there was also significant effects on the ecosystems on the island as well. So I want to take you back to approximately a year and a half before the hurricane. I had visited the island during May of 2016 because of the Zika outbreak that was going on at the time there and throughout the Caribbean. And I went to the El Verde field station, which I want to talk a little bit about coming up. And at the field station, I shot this picture, and this was what the forest looked like at the time. It had been about 20 years since a major hurricane. And what you notice is that the canopy is relatively solid. In other words, there's not a lot of light gaps, and there's essentially no understory. So you can see the boulders, you can see down trees, etc. You don't see a lot of vegetation. The hurricane hit in September of 2017. I went back in January of 2017, and this is what the canopy, 2018. This is what the canopy looked like at the time. You can see most of the canopy had been removed, and that ended up on the forest floor. So what these hurricanes often do is deposit the canopy on the ground, essentially. Now, fast forward about a year and a half after this, and we see that we now have what's known as an inverted forest. Most of the biomass, the living biomass, is now on the ground. So herbaceous plants, weeds, and other non-woody vegetation kind of take over because of the amount of light infiltration due to the lack of canopy. It will take the canopy years, if not decades, to recover. Many of these trees are very long-lived, and they're not being replaced fast enough because the rate of hurricanes has been accelerating because of climate change. And so what we now have is this inverted forest in which much of the biomass is on the ground. So some of the questions that we were interested in on the island were, what is the effect of this movement of materials from the canopy down to the ground? What is that likely to do to insect communities? And specifically, we wanted to focus on insects, specifically mosquitoes, in the capital, San Juan. The reason that we wanted to do that is because this is one of the few places within the United States that you can actually go and potentially get dengue, chikungunya, and Zika. And in fact, there's all 4 serotypes of dengue, so you could actually get dengue 4 times if you were so inclined. So this makes it a unique opportunity for medical entomologists like myself to study circulating pathogens as well as the mosquitoes that vector them. And back in 2016, this was before the hurricane hit, we had done a project that was focused in an area in San Juan known as the Caño Martin Peña. The Caño Martin Peña is essentially an estuary that flows from the San Juan Bay in the west out to a number of forests and marshes to the east, passing through several neighborhoods. You can see this is the airport over here if you've ever been on the island, that can sort of orient you. So this is where the bulk of inhabitants live in San Juan. This area along the Caño Martin Peña, though, is rather unique in the sense that the residences and the housing that's along those areas are considered very low on the socioeconomic scale. So if you were to go back and look at this particular area about 80 or so years ago, if you were to look at the map, so this is the Caño Martin Peña again flowing through this area, what you notice is some developed neighborhoods, and then you start to see these little fingers coming down. What these fingers are, these are non-planned neighborhoods. These are essentially like a shanty town or other kind of low-income housing that was erected because these individuals had nowhere else to go. So they just started building their own houses and just putting them wherever there was space available. And that space that was available, you know, is right along the river. And for those of you who are ecologists who know something about rivers, you know that the area on either side of a river is called a floodplain. And the reason it's called a floodplain is because it floods. So this area never had a planned system, and therefore the housing that's along these areas experiences these annual periodic floods due to storms or other coastal events. And what happens is that this area also doesn't have any infrastructure in terms of things like sewage. So most of the houses then release their effluent into the Caño Martin Peña. When these storms happen or these flooding events happen, that water then comes back into those neighborhoods. These neighborhoods are also home, as I'll talk about in a second, to the major vector of all these infectious diseases, and that's Aedes aegypti, the yellow fever mosquito. The other thing that's very common along these neighborhoods is garbage. Again, lack of infrastructure has led to a lot of garbage just being left around. This is behind somebody's house. This is bags of garbage that are essentially just floating in the canal because of that. And as we'll see, this leads to a lot of opportunities for mosquito production. So this nitrogen then that is part of human waste is coming back into these neighborhoods. And in 2016, we were hypothesizing there actually may be a link there to the production of Aedes aegypti, the yellow fever mosquito. So just as an aside, I think this is one of the best graphics ever made to illustrate human mosquito interactions. This is actually from an NPR story done back during 2016. And what it shows is an individual who is walking probably somewhere in the Caribbean or in South or Central America. You can see that it's a relatively low economic condition. You can see that there's no screens on the windows. You can see also that there are lots of places for these mosquitoes to rear their larvae. And Aedes aegypti, which you can see here, the yellow fever mosquito, will use things like buckets, small containers, and tires as a place to rear its larvae. The other thing is this is a very human adapted species. This is one of the most human adapted species that we have across the world. They live in houses, especially if there's no screens. They take blood meals almost exclusively from human beings, and they like to lay their eggs in containers that have human association. They're also adapted to bite you on the backs of your legs and on your ankles because that's a place where you're less likely to swab them. So Aedes aegypti is a really important vector then for these various arboviruses. And this neighborhood specifically has lots of containers that you can find laying around that have the production or potentially that produce aegypti. And there's also lots of potential hosts. So this is one of our mosquito traps here on someone's porch, and you can see they have a cage of chickens. There are also lots of stray animals, including things like pigs, and also a lot of invasives like iguanids. So in 2016, we devised a project to investigate whether or not the influx of this nitrogen would affect the production of Aedes aegypti and also Zika production. And so what we did is we located neighborhoods that were in an association with the Caño Martin Peña, as also areas that were outside of that. In each of the neighborhood, we took water samples from the environment to look at the amount of nitrogen, both in terms of percent nitrogen as well as the stable isotopes. We captured adults. We also captured larvae and analyzed them for their stable isotopes. And then those adults were also assessed for the presence of Zika. And this was the Caño Martin Peña neighborhood that we focused in on. So we made this heuristic diagram that was basically meant to show how these various sources, of nitrogen could influence various aspects of the life cycle of the mosquito, but also in terms of the potential for disease transmission. And just kind of real briefly, I just want to go over some of these. This was actually published a couple of years ago in EcoHealth. But some of the results that we found, for instance, for nitrogen, is we found that the amount of leaf material, the amount of leaf detritus was influencing the amount of nitrogen that was found in the containers. And these leaf environments or these leaves actually provide a lot of resources for developing larvae. We also found that among these neighborhoods, the amount of nitrogen in these containers influenced in a positive way the amount of nitrogen that was found in the larva. And then we also found that the amount of detritus that was found in leaves in these environments was also influencing the amount of nitrogen in a positive way that was found in adults that we captured in those same neighborhoods. And then finally, one of the more interesting things is that we found that the amount of nitrogen that was found in adult mosquitoes across these neighborhoods was actually influencing the number of copies of Zika virus that those individuals had in their body. So we find that the nitrogen that's in the environment influences not only the production of these mosquitoes, but potentially the production of those mosquitoes, or sorry, especially in relation to their ability to vector a particular disease. So we had already done quite a bit of work in some of these neighborhoods in Puerto Rico. And then in 2017, Hurricane Maria hit. So I applied for and was lucky enough to receive a rapid grant from NSF to explore how changes in the resources based on the hurricane would affect the production of these mosquitoes. So we had a variety of research questions in 2017 and 2018. First, are there spatial and temporal effects of Hurricane Maria on populations of Aegypti? And we hypothesized that because the hurricane disrupted nutrient inputs, that would have an effect on the adult production, and that we assumed that that disruption would not be equal across neighborhoods, because higher socioeconomic neighborhoods would be better able to withstand those hurricanes that have better infrastructure, whereas the low socioeconomic neighborhoods would not. We also asked whether or not there was spatial and temporal effects of Maria on nutrient stoichiometry, specifically carbon and nitrogen ratios within the mosquitoes, their nutrient content, and their stable isotopes on females specifically. And we hypothesize again, because the hurricane likely disrupted those nutrient dynamics, would it have short and long-term effects on those mosquitoes? So we sort of expanded our sampling from 2016 to look at 8 neighborhoods that varied in socioeconomic variables. In each of these neighborhoods, then, we placed out 6 BG sentinel traps, which are an adult trap to catch host-seeking mosquitoes, as well as looking for containers that contain larvae. We did this sampling in January, March, May, and July, and October of 2018, which would be the year after the hurricane, and then 2 months in January and May of 2019. And in each of those cases, we were looking at the number of females that we caught in each neighborhood, the percent carbon, nitrogen, C to N ratios, and stable isotopes of the females, as well as the larval surveys. I should just mention real quickly that in July of 2018, American Airlines lost all our baggage, and therefore we lost all our samples since we were transporting them in baggage. And therefore, I don't have that data to share with you. We do have the female abundance, but we don't have any of the stoichiometric data. And if I still sound bitter after 3 years, I am. So these are the neighborhoods based on socioeconomic status. I won't go into the details of how we computed this, but needless to say, these do vary statistically significantly. So we had 3 low socioeconomic neighborhoods: Catano, Martin Peña, which I've already talked about, and Torresilla. We had 4 that were sort of middle income or middle socioeconomic status: Puerto Nuevo, Rio Piedras, El Comandante, and Vista Mar. And then we had 1 high socioeconomic location, Villavencia, which is a gated community where people have sports cars and speedboats and things like that. So our sampling was pretty similar to the 2016 work, except for we were specifically focusing in here on females. We were taking female heads once we captured them in BG sentinel traps, put them in RNA later, and then froze them to do viral analysis, which I'm not going to talk about today. And then the bodies were dried for nutrient analysis and stable isotope analysis. Okay, so what are some of the results that we found? Well, in terms of abundance, what we found was a significant date effect, neighborhood effect, but no date by neighborhood interaction. So I'm just going to show you these independent effects. So first of all, this is the date effect. So this is January after the hurricane. And then this is over here, January, a year after the hurricane. And what you can see is that there's no significant difference in the number of female mosquitoes across these times, except for this dip in March that was likely due to the dry season. So dry season in Puerto Rico is usually at the beginning of our year, sort of our winter. And then the rest of the year is the wet season. So there was no effect of Maria on the production of mosquitoes. Now, again, the hurricane was in September, so it's possible that more mosquitoes were produced in the months or two after the hurricane, but those may have decreased by the time we actually got our money and we were able to get down there and start sampling. As far as the neighborhood effect, what we found is that lower SES neighborhoods tended to have more adults. So Torcia, which is one of our low socioeconomic neighborhoods, was significantly different. I'm sorry, this should be an A right there. Should be significantly different than all of our middle and high socioeconomic. And then two of our other low socioeconomic neighborhoods were intermediate. In terms of nitrogen and other nutrients, what we found was a neighborhood effect, mostly explained by nitrogen and delta nitrogen, which is the stable isotope. So when looking at this across our three neighborhoods, what we found is that typically females that were in our low socioeconomic neighborhoods here in yellow tended to have slightly higher, in some cases significantly so, nitrogen, delta nitrogen, which is again related to human inputs compared to other neighborhoods. And this is reflective of the work that we did in 2016. And then if we look at percent nitrogen, we're kind of seeing the same thing, at least in one of our neighborhoods, Martin Pena, which I already talked about, significantly higher than our gated community, our high socioeconomic neighborhood. Now, these two neighborhoods, one of the interesting things about them is both of them flood. So both of these neighborhoods, as I shown you before for Martin Pena, as well as Catania, both of these potentially are receiving those inputs of nitrogen through sewage when it floods, and therefore that may help to explain why they have higher delta nitrogen. Okay, so we generally have higher nitrogen values and lower SES, and that could be related to flooding. We also found a date effect. The date effect is interesting. It's really hard to discern any patterns when we look at female mosquitoes in terms of their percent nitrogen or percent carbon. We can see some patterns, for instance, in nitrogen. We get this set of months that are grouping, but they don't really make sense in terms of the time of year. We can also do the same thing for carbon. So we can see that there's a couple of months here that seem to have higher carbon than one of these other months. It's not really until you look at carbon nitrogen together, because they're being allocated in tandem with each other within the mosquito itself, that you really start to see some interesting patterns. And this was a pattern we were really hoping to see. So in January of 2018, our mosquitoes had significantly lower carbon and nitrogen ratios compared to a year or a year plus later. And that indicates to us that there was something different in the months following Hurricane Maria compared to a year or more after Hurricane Maria. And we feel as if there's something about the nutrient dynamics that had changed at that time, because we don't expect to have this large of a variation in CN ratios among these adult females. So we think what we have here is a signal of Hurricane Maria that manifests itself a few months after the hurricane, but then is gone by a year or so later. So for our research questions, we found spatial and temporal variation in populations, but they didn't seem to be reflective of Maria. They seem to be more reflective of these differences in socioeconomic status. In terms of distributing nutrient inputs, we really didn't see that for Maria. We did again find differences in nutrients across different neighborhoods. Our second question was about spatial and temporal effects on nutrient components specifically. And here we did find that C to N is likely to have changed due to Maria. And we also found neighborhood effects as well. And that we did find support for the fact that nutrient inputs across neighborhoods were likely due to Hurricane Maria. So overall, what we find is maybe not subtle effects, but we find effects on the mosquito itself that are due to this hurricane that may have nothing to do with its ability to vector disease. Okay, for the last part of my talk now, I want to switch to some other work that I do in Puerto Rico, and this is specifically with relation to the Laquillo LTR. If you're not familiar with LTRs, long-term ecological research sites, there's about half a dozen or more of these that are established around the United States and in the Caribbean that are meant to give researchers a longer time to collect data. Typically, we, as either graduate students or even faculty, are on multi-year data sets at best, whereas LTR sets are often on decadal timescales. The Laquillo LTR was actually established back in the 1980s and has been in continuous operation and has been collecting data ever since. So it gives you this really long-term data set. So I'm going to take you now from the city over here in San Juan over to El Verde Field Station, which is the field station centered within the Laquillo LTR. the Liquillo LTR is actually located in El Yunque National Forest, which is the only tropical forest under U.S. Forest Service jurisdiction. So the LTR has, like I said, been there for about 40 to 50 years. It has research infrastructure. There are laboratory facilities and dormitories that are available for scientists to use. And this location did get hit pretty hard by the hurricane. So you can see on the left here, this is one of the research labs before the hurricane. This is what it looked like after the hurricane. And in fact, much of the field station still has considerable damage, although it is functioning because of the hurricane back in 2017. Okay, the reason I got involved in the LTR, I was actually a graduate student. I did my master's work in the LEF, not on mosquitoes, but on aquatic insects specifically. And in 2016, when I visited the island to start working again on mosquitoes, or working for the first time on mosquitoes, I was able to find myself a home at the LTR as well, working on the ongoing research, in this case, specifically for litter invertebrates. So these are insects that would be found in the leaf litter in the tropical forests. So the LTR started new sampling in 2017 along what's called the LTEP, the long-term elevation plots. These are plots that range from 300 meters up to 1,000 meters at every 50-meter increment along two transects, a palm transect, which is the P, and a mixed forest transect, which is the M. The idea was to do these every 6 years, so 2017, 2023, 2029, et cetera, and use that to investigate questions related to climate change and disturbances. Well, what none of us knew is that the first year, 2017, would be followed by a hurricane. So all of the work was done during June, July, and August, and then the hurricane hit in September. So many of us decided to, instead of waiting for 6 years, to go back in 2018 and sample these transects again for to see what the effect of disturbance was on these organisms. So at each of these elevations, which are called strata, there is a sampling plot that looks like this. It's 20 by 50 meters. It's all laid out in a grid. And this is available then for various researchers to collect data on a wide variety of taxa. That's the goal of this, is to collect as many aspects of the food web as possible. So you can see that in the center, people look at things like plants, seedlings, decomposition. There are places where snails, which is the dominant herbivore in the forest, are sampled. There are soil biogeochemistry that's taken. There are data taken on birds, amphibians, pretty much anything that you can think of. You can also see down here in this bluer color litter invertebrates. So what we did is in each of these strata, we took 10 litter invertebrate samples. These were half-meter quadrats. So that gave us within each strata 10 samples. There were 15 strata in mixed forest, 15 in palm forest, that total of 300 samples. For today's talk, I'm only going to focus in on the mixed forest, so that's 150. And we sampled these in 2017. And then we went back in 2018, as I mentioned, and we sampled these again to look at what the effect of Hurricane Maria was on the litter invertebrate community. So the sampling protocol was to take the litter, place it into cloth bags. They would then be taken back to the field station. We'd use a passive extraction system known as Winkler samples, wherein we would sift the bulky litter. We'd place the fine litter and all the invertebrates into these bags. Over the course of 2 days, the invertebrates would move out of those bags and move towards the bottom because that's their natural behavior. And they would end up in these collection bags here of alcohol. Those would then be sorted to the lowest taxonomic level possible, usually either order or family, and then we could look at the data. So what were our results? So we looked at total number of individuals as well as richness. Now, in this case, this is ordinal richness, so it's not species richness per se. And what we found in all cases between our 2 years is an elevation effect, a year effect, and an elevation by year interaction. And generally in 2017, we had significantly fewer invertebrates compared to 2018. We had about a threefold increase in the number of invertebrates per sample, and the number of species, quote unquote, again, this is ordinal, went up slightly. So what did this actually look like? So when we look at total abundance in the dark symbols here, this is 2018, this is post-hurricane. in the open circles, this is 2017, this is pre-hurricane. And what we found is significant increase generally at lower elevations across these. We also got a bump at 650 meters as well. We didn't see any other differences at these high elevation sites or some of these intermediate sites. And in terms of richness, we didn't have enough statistical power to determine differences, but it looks likely, again, that richness likely increased at some of these lower elevations. How about particular taxa? How are they experiencing this? Well, if we look at all these groups, again, darker is going to be post-Hurricane Maria, lighter is pre-Hurricane Maria. What we find across the board are positive effects of Hurricane Maria. In general, insects went up about 300%, non-insects about 350%. And if we look specifically at some of these groups, we find that there was a compositional shift as well. So pre-hurricane, the insects or the litter invertebrates were dominated by ants, which is here in white, along with Collembola, Diptera, and a few other groups. And after the hurricane, you see the abundance of ants went up significantly. Some of these other groups kind of went down a little bit or didn't increase as much. So the only group that actually went down were our non-Hymenoptera ants, but you see the ants overall went up 500%. Coleoptera, the beetles, went up 200, Collembola 330, and arachnids went up 220. And I'm going to focus in on these four groups to look at those same patterns in elevation and year that I mentioned before. Okay, so again, significant effects across the board. When we look at ants, we generally find that same pattern as we did for overall abundance, in that lower elevations seem to benefit from the hurricane. When we look at Collembola, again, went up significantly at lower elevations. For Coleoptera, lower elevations again were benefited. And then for arachnids, we actually had both low and intermediate elevations. We saw an increase after Hurricane Maria. So in all cases, we're getting more insects at those lower elevations, not so much at the higher elevations. And that's really interesting, but this is only part of the pattern, because what we're not talking about yet is species level identification. These are just all grouped together. So we're going to take a deeper dive onto the ants. And this has only been possible over the last year or so because we've been able to get species level identification for all of our ants from these 2 years. And this was really with thanks to two folks, Jack Longino and James Wettner, who did all the taxonomic stuff for us. And for this project, then we wanted to know, did ant richness increase? Did it change in conjunction with total abundance? Did occupancy change? In other words, the number of these samples that were occupied by particular species? And did we find compositional changes as well? Now, one of the caveats for working with ants is that they're social, which means that some of the analysis that we would typically apply to animals that are independent, so like a bird or a frog, which is an independent organism, don't really apply. So there's some things we can't do, but we can still look at year-to-year changes across the plots or within a plot to get at some of these answers that we're interested in. So the first thing we wanted to look at is differences in ant richness. First, I should say we ended up with 27 species of ants between the 2 years. And what we found is no significant difference between elevations within the same year. So we did find a year effect. We did find an elevation by year effect. But when you look at, for instance, 400 meters, you don't see a difference between those two in terms of the richness. You don't find it for 600, et cetera. There are some differences. For instance, 450 is significantly different than in 2018 compared to 2017, but that's not really a relevant comparison. So we don't really find differences in richness. So richness didn't really change between years within an elevation. How about occupancy? Again, this is where we're looking at whether or not an animal is present or absent, a species is present or absent in a particular sampling place across those 150 sampling locations. So what we did is we did logistic regression on the 9 most common, and the 9 most common ants here represent about 97% of all individuals collected over that time. And you can see they vary in terms of their ecology. Some are native, some are more Caribbean widespread. Most of them, though, are found in the litter or in rotting wood. We had one here that's arboreal, but still is found on the ground. And what we find is that in all cases, we get no year effect. So individual species aren't seeming to benefit from pre and post Hurricane Maria. But we do see elevational effects in occupancy, something like this. So this is Wasmannia. This is a widespread, potentially invasive ant in Puerto Rico. And what you see is that it's more likely to occupy these lower elevations than, for instance, these higher elevations. So even though we saw an increase after Maria in the numbers of individuals, we're not seeing it in terms of occupancy. They're not expanding their habitat due to the hurricane. They're kind of staying where they were before, but they're just getting more of them. And then finally, were there compositional changes? The way we approached this was to do a principal component analysis on those 9 most common species. And then that reduced those 9 species down to 4 factors, which explained about 60% of the variation. And then we used those factors in a multivariate analysis of variance. And what we found is significant elevation by year effect. So the first factor that was important was factor 1, and that contained these 3 species. And what you can see is that these 3 species increased quite dramatically at low elevations, which is something we've already heard before. For factor 2, it was Philadolia and Solenopsis. They increased also most significantly at this lower elevation. And then factor 4, which contained these 2 species, also increased at this lower elevation. So it's a broken record, but putting this all together, we're starting to see that there is something different about these low elevations in terms of their effect on ants. So in our conclusion for this project, we did find an overall decline in ant richness, but not due to elevation. In other words, we didn't find those elevational changes to be consistent across the 2 years. Occupancy did not change, which suggests again that these ants aren't really expanding their range across the elevation. They're sort of hunkering down where they have always been. Compositionally, we did find that certain taxa benefited from the hurricane, but again, only to increase their numbers at these lower elevations. So in general, for all these analysis, for all this blood, sweat, and tears, so to speak, what we're really finding is that the increase in abundance is really at these low elevations. And the question is, well, why? What's unique about these low elevations? Right now, we don't know. Right now, we have the pattern. We need to get into the process. We need to figure out why this is. One hypothesis is it could be that these lower elevations experience greater inputs of detritus after the hurricane. So lower elevations may have had a more complex canopy, and therefore after the hurricane, more of that ended up on the ground, which was going to be available and beneficial to these particular ants. It also could have been that there were microhabitat changes. So it could have been that those at the higher elevations, since they were already sort of depauperate in terms of the amount of nutrients, they had even lower nutrients, where those nutrients may have moved downhill after the hurricane, and that may have boosted what the available resources and some of the microhabitat changes that were available. One of the things, though, what we'd really be curious to know is what is the temporal perseverance of these trends? In other words, did the changes that we saw in 2018 persist? Well, unfortunately, we didn't take data in 2019 on the elevational plots. We actually switched to a different sampling protocol or a different sampling location. And I'm going to introduce you to that, tell you a little bit about it, and then I'm going to tell you how I use that data to answer this question about persistence. So back when the LEF was established, back when actually I should say before even that, when the Fish and Wildlife Service the Forest Service purchased this property and made it into the Keogh Experimental Forest back in 1934. About 50 years later, the folks who are working at the LTR established what's been called the Big Grid. So the Big Grid, also the Keogh Forest disturbance plot, is a 16-acre hectare plot that contains within it 3 historic legacy disturbance regimes. And those disturbance regimes are more or less equally represented. So on the grid itself, there are 442 grid poles. These are 1-inch PVC poles that have been driven into the ground. They are laid out, there are 440 of them. They're laid out in a massive grid. The larger poles are 20 meters apart in any cardinal direction. Smaller poles are shorter, and they're every 5 meters in between. It's very difficult to get lost because everything's labeled out there, even though it's a quite dense forest. And the 3 historic land use are based on canopy cover, based on data from 1936. So category 1 to 2 was 0 to 49% cover. This was historical property that had been clear cut. Essentially, all the vegetation had been removed, and that's why you get 0% cover. Category 3 was between 50 and 80%. This was mixed agriculture. This was generally coffee plantation. So much of the canopy or some of the canopy was maintained, and then coffee shrubs or bushes were planted underneath them. And then we got category 4, which is 80 to 100%. This is just areas where they would go in and take out a mahogany or take out some sort of big tree and leave everything else in place. And that's why there was relatively high canopy cover. The big grid also exists across an elevational dynamic as well. And you can see that the southern part of the grid is at about 400, 430 meters. And then the lowest part of the grid is just around 200 and 250 meters. There are two streams that flow through it, the Toronja, which is grapefruit, and the Prieta. And then these other lighter lines, these are trails that actually lead out there. So these categories then, as I mentioned, have been replicated, or I should say the points are out there that have been replicated through time to represent these three. So the darker is your category 1 and 2, your lighter is category 3, and then the white is category 4. So what we did is in 2019, we wanted to know whether or not there was any kind of legacy from these historic land use patterns on the litter invertebrates. So what we did is we took relatively similar number of samples in these different land use categories or historic land use categories, giving us a total of 120 samples. We took these in the exact same way that we took the elevational plots, that is half-meter quadrats, which the litter was then placed into bags, processed in Winklers, and then lowest taxonomic level possible. So what did we find? Well, first of all, we collected about 6,000 invertebrates. And just like with our elevational plots, the dominant group were our ants, followed by termites, spiders and their allies, arachnids, as well as millipedes. And those four really constitute the vast majority of insects that were on those, within those 120 samples. And nicely, these also fall along different food web or different places within the food web. So we have predators and fungivores in the ants, we have herbivores with our termites, we have predators, and then we have detritivores with our millipedes. But what did we find in terms of those patterns that we were looking for? First of all, we did not find a significant effect on those historic land use on either richness or abundance. So again, we got about 50 animals per sample, about 6 quote unquote species. Again, these are mostly at the ordinal level. So there didn't seem to be a long-term effect of these historic land use types on the litter invertebrate communities on the big grid. How about for elevation? Nope, we also looked at elevational effects. There were no elevational effects. So what we see is that the insects seem to be somewhat homogeneous across the LDF. However, we can dig deeper into some of these major groups, and we can look at to see how they responded. And we get a slightly different answer. So when we look at the taxa by these groups, whether it be class or lower, actually not class, I should say, by order or lower, in some cases class, what we see, this is our metric for disturbance here. So this is our 0% canopy clearance. This is 80%. So this is essentially fully disturbed. This is almost no disturbance here. So that's what this triangle represents. We can see for our millipedes, essentially there's no difference across those three. They don't seem to really respond to those historic land use types. However, our ants do, and our ants are much more likely to be in low abundance when there has been low disturbance. So they really seem to be disturbance tolerant, or maybe even benefiting from disturbance, even though that disturbance took place 80 years ago. So there's still this echo, right? Arachnids have this U-shaped response. So they tend to be lower and moderate amounts of disturbance, but higher or not significantly higher, I should say, compared to the lowest level of disturbance. And then isopods show kind of an opposite with our ants in that they're much more likely to find in low disturbance environments as opposed to high disturbance. I should note that this was not significant. That's why it has 3 A's, but it was approaching significant with 0.08. So the reason I've shown you this data is, number one, I think it's cool, but more importantly, we can actually use this data to help us fill in the gap about persistence of ant communities after Hurricane Maria. So what I'm going to do is I'm going to put together the mixed forest transect and the Lichio forest dynamic or disturbance plots. And when I do that, this is to remind you of what happened with our ants during 2017, which is before the hurricane. 2018 was after the hurricane. We get a significant increase. Now I lay upon it the data from 2019 at the LFDP. And what I should say is that, remember, these are across 700 meters. These are only across 200. What I've done here is I've truncated so they're all at the same elevation. So everything you see here has been collected at the exact same elevation in the exact same forest type. And what you see is that no matter what historic land use type you look at, they are significantly lower compared to the hurricane, suggesting that this effect of the hurricane was very short-lived, perhaps a year at most. Now, my students and I just got back from Puerto Rico less than a month ago, about 3 weeks ago or so, and we took the LFDP data for this year on the big grid, and hot off the presses, this is what it looks like this year. And we can see that even though I haven't analyzed it yet, we can see that it's going to be similar to the past few years in terms of the amount of ants that are there. So this is definitely a hurricane effect, and it does not seem to persist past that. So overall, what we see for ants, for termites, for some of these other groups, there is an ecological echo. Even though this has happened, 40, 80, 100 years ago, they're still showing signs of this. That's probably related to the forest that grew up in those areas, but that the hurricane effects seem to be short-lived. All right, so for our future directions for this work, in 2023, we plan to take our elevational plots again, and in the intervening time, we're going to do the LFDP. 22, we couldn't take any data because of COVID. Everything got shut down, unfortunately. And we want to relate these populations, specifically ants, to things like plant composition and biomass and soil type. These actually do exist. Again, this is an LTR site, so there's lots of data out there that we can mine to look at perhaps what these communities are responding to. And we actually are proposing that ants are likely a key disturbance group, not only in the short term, as we've seen with Maria, but also in the long term. And these are one of the groups that responded to those sort of legacy disturbance regimes. We have about 30 species. We have the pre and post LTR. And now the next thing to do is to look at more compositional changes. I see my time is almost up, but I just have one more project to share with you. I can't help but getting one more thing in there. And this also relates to disturbance, but this is an entirely different kind of community. So this is a community that exists in Puerto Rico, found within a plant called Heliconia caribaea. So this is the plant here. You see these rather large inflorescences, and these inflorescences are actually termed a phytotelmato. So the plant itself is a gap specialist. It only grows where there's sufficient light. So along riversides, where there's been a tree fall, etc. And the clusters of individual bracts here form what's called a raceme. And these actually hold water. So if you can kind of look at those closely, what you can see is there's actually water in these little cups. And those phytotelmata are actually home to more than 20 insect species. And these are predominantly aquatic diptera, but there's a few other taxa in there as well. And they form meta-communities. So essentially from plant to plant, from raceme to raceme, you get an individual community. There's also temporal dynamics because the ones at the top are younger than the ones at the bottom because they're produced sequentially, etc. So there's all kinds of really cool questions you can ask with these systems. So this is one of the animals. These are the large rat tail maggots. These are syrphid flies that will eventually turn into bees mimics. And I did some stable isotope work in the system a few years ago, just to kind of show you what some of the variation in things like the amount of nitrogen and carbon is among these taxa, as well as some of the flower parts. So the yellow are actually different portions of the flower. The diamond is actually the fluid that we find, in the environment. The circle is actually snail feces, which is very common in these heliconia. And then the black, gray, white are all different taxa. And what you can see is a wide variety. This black one here is Toxorhynchites, which is a predatory mosquito. That's why it's on the far right side here. It's been enriched in nitrogen by eating everything else that's in there. So it's a wide, really cool food web, and also really cool in terms of the invertebrates. Well, one of the things that we were interested in is how do these communities respond to disturbance, both short and long term? And what do you do when plants are knocked down after a hurricane? Because that's typically what happens. I had an undergraduate, Jacqueline Everly, who in 2017 did a couple of projects for her honors thesis at the University of Southern Mississippi. And one of the projects was to look at whether or not we could simulate these heliconia, because after a hurricane, the plants would get knocked down. We couldn't assess whether or not that community of insects was still viable within the forest. So what she did is we kind of came up with an artificial system here to replicate the heliconia overall. And so she replicated two sizes, those racemes that had either 5 bracts or 8 bracts. She filled these containers then with an appropriate amount of liquid that you would find in the heliconia bracts, along with dilute sugar, which is also found in the bracts because they're nectar producers, as well as some detritus. And then she left these out to be colonized by invertebrates to see whether or not we get the same community. Spoiler, we did. And she also collected these through time at 2 weeks, 4 weeks, and 6 weeks after establishment to see what the colonization pattern is. I'm not going to go into that data, but what I do want to talk to you about is differences in the communities, because not only did Jacqueline take this data in 2016 with the odd chance that there might be a hurricane some point in the future, but because there was a hurricane literally 3 weeks after she left the island, we were able to replicate her experiment in 2018 with a different group of students. So we have before and after hurricane data, just like we had for the litter invertebrates. And we're going to look at these both in terms of alpha and gamma diversity. In this case, alpha diversity is the average diversity that you find within one of these cups, whereas gamma diversity is the sum across the entire cluster and the entire group of individual habitats. So based on analyses, what we found was a significant year effect, both in terms of alpha and gamma diversity. What did that look like? In terms of alpha diversity, we found significantly fewer individuals, went from 90 on average to about 25, and species from about 5 down to about 3 after the hurricane. So these heliconia communities were hit and they didn't seem to respond very well. At the gamma level, so this is looking at the entire racine, we went from about 500 individuals in an entire racine down to about 160. So massive decline in number of individuals, and then a decline as well, a significant decline, not only in species richness, but an increase in evenness. So we found higher evenness after the hurricane. Now, this was her experiment. She also took data on actual heliconia communities. We weren't able to replicate that as well after the hurricane, but we took enough data to make a comparison. And what you see is here in living heliconia, before the hurricane, those are the open circles here. We have replicates that are with 5, or replicates that are in the squares here with 8 racines or 8 bracts. And you can see richness versus abundance. And then after the hurricane, the ones that are in solid. And generally what you see is that before the hurricane, we have relatively higher richness and higher abundance compared to after the hurricane. So although we found increases in our ants and other litter invertebrate communities, we actually found a dramatic decrease in these heliconia systems. So overall, for the forest work at least, we can conclude that there was a positive influence of the hurricane on the litter invertebrate. And perhaps that was because it was increasing the litter and benefiting the habitat in terms of moisture and food availability. We don't have a good handle on all the compositional effects. We do know ants changed a bit. And we also don't know whether or not some groups benefited because they have traits that are more in line with disturbance. And that's something we want to look at in the future. In the forest itself, in the canopy, and in these heliconia, which grow within the forest itself, we found generally negative effects. And those may be because the forest became generally drier. There was much more light. And there may have been less resources available for these insects to colonize the heliconia. And that gives us a question about whether or not there's an impact on long-term stability of this meta-community. So I think with that, I'll go ahead and end, and thank you. And at that, I am very happy to answer any questions you might have.
Fantastic. Thanks, Don. Really appreciate it. We do have a few questions. So Anna asks, Aedes aegypti is introduced from Africa, correct? Are there insects or other organisms that are native to Puerto Rico that have become well adapted to humans or urban environments?
I would say no. So if you go into the urban environments, especially into some of the lower socioeconomic neighborhoods, you do see a lot of invasives. So I showed you pictures of iguanids, which have now become established on the island. Those are not native. Your typical pet store iguana, the green iguanas, those are kind of all over the place. They're not established. We also have cats, dogs, rats that are relatively common. Pigs are common. So in the urban environments, at least, we don't see the establishment of native species, if you want to call them that, endemics. There really aren't that many endemics in Puerto Rico. There's one bat, there's a few birds, there's likely insects, but they're not very well studied. And often what we see in Puerto Rico are species that are often widespread among the Caribbean islands. So Aegypti likely got to the Caribbean in general 400 or 500 years ago because of the African slave trade. And it is ensconced on pretty much every Caribbean island with human populations.
Rachel feels for your tragic loss of samples. Not a question, but I thought I'd share that sentiment.
Did I mention it was American Airlines?
I think you did have a logo.
I think I may have, yeah.
Okay, so Ben asks, have you observed the displacement of native mosquito species in container habitats by invasive ones like Aedes aegypti?
Right, so this kind of, I think this kind of goes back to the first question a little bit. Again, not so much. I can kind of bring you up to date on the invasiveness on the island. So at the present time, there is no Aedes albopictus. Aedes albopictus, the Asian tiger mosquito, has seemingly not been able to invade Puerto Rico, even though it is in the Dominican Republic, which is about 200 miles west, and it is on some of the other major islands like Jamaica and Cuba. We don't know why it hasn't been able to invade. We just did a survey. This is the crowdfunding that Katie mentioned before. We crowdfunded to do the mosquitoes of Puerto Rico, which was just published earlier this year. And we spent 3 weeks going around the island and we didn't find any albopictus anywhere. So we don't know why. One, there are a couple of hypotheses for us. One may be it has to do with trade. The way that albopictus historically has been moved around is through the tire trade. And one of the things that Puerto Rico is a little bit different is that Puerto Rico is only allowed by treaty to trade with the United States. So foreign entities cannot bring their ships into the port in San Juan and offload material. It has to come first to the United States and then it can come. So that may be somewhat of a barrier to bringing in albopictus. However, we are trading with mainland US and that does have albopictus. So that may or may not hold water. One of the other ones I can briefly talk about is a native mosquito on the island called Aedes mediovittatus, which is the Caribbean treehole mosquito. And for those of you who are well versed in Katie's work or in Steve Giuliano's work, one of the things that's interesting is that when Aedes albopictus invaded the United States, it's thought that Aedes triceriatus was able to some ways resist its expansion into forests, either because of the presence of Toxorhynchides, the predatory mosquito I mentioned before, or because of some ability it had to better cope in those environments. Well, in Puerto Rico, there is a Caribbean treehole mosquito, Aedes mediovittatus, and it is found in more rural locations, which are probably more amenable to what we find albopictus invading in other places around the world. And so it may be that it's simply niche preemption. Maybe mediovittatus is already there and is doing the things that albopictus would do if there was no albopictus, right? So that might be another, and we're actually exploring that. We've already done some competition experiments and we're working through those as well to look at mediovittatus. And then kind of the other side, this is for your horror story reel. There's a new mosquito in the Caribbean called Aedes vittatus, not mediovittatus, but just vittatus. That has recently shown up on the Dominican Republic and Cuba. It's probably established in both of those locations. And it may also already be other places. People just haven't found it yet. And this is a mosquito that is native to Africa, but it's expanded into Europe. It's in Spain. And it is a container mosquito that is a competent vector for dengue, chikungunya, Zika, and other things that albopictus and aegypti are. So it's another clone of these two invasives, and it may show up in Puerto Rico at some point. I wouldn't be surprised if in the next 2 years or so it's going to be on other islands.
So Riley asks, did you see a difference in the carbon and nitrogen levels in different neighborhoods? And any guesses on what nutrients caused that carbon and nitrogen difference?
So yes, so one of the things that we did find is that our lower socioeconomic neighborhoods often have greater nitrogen and greater delta nitrogen. So I didn't really fully explain that. So the stable isotope that we use for nitrogen is delta 15. That is the one that is often related to human sewage. So if you look at wastewater treatment, they're often measuring delta nitrogen 15. And you can see that signal then picked up in anything that eats that sewage or is a byproduct of that sewage. So generally what we found is those higher delta nitrogen values in lower socioeconomic neighborhoods. And we think, and this is something that I talked about very early, is that we think it's this flooding events. When this raw sewage is coming back into these neighborhoods during these flooding events, and then it recedes, it gets left in those containers. So all that trash that's laying around, those tires and those buckets and everything else, are initially going to have that sewage water in them, but as the water recedes after these floods, it's left behind, and then the mosquitoes colonize and they take advantage of it. The other possibility is that that is getting into nutrients such as detritus in plants that is then making its way into the containers, which serve as a food source for the larvae. So there's a couple of potential mechanisms there. And these moderate to high socioeconomic neighborhoods just don't have the flooding, and they're much more manicured in terms of the vegetation that they have around.
What do we know about the carbon nitrogen content of adults and how that translates into longevity or fecundity?
So as far as fecundity and longevity, typically mosquitoes that are grown on high nitrogen diets, such as an animal-based source. So if you grow them on dead crickets or give them something like bovine liver powder, what you find is that they're bigger and they live longer. We did a project a couple of years ago with somebody Katie knows, Barry Alto at the University of Florida. And what we did there is we grew mosquitoes, Aedes aegypti, on various levels of nitrogen based on their diet. And then we looked at their competence for Zika. And what we actually found there is that the more nitrogen that they got as a larva, the less competent, the less Zika that they had as an adult when they were offered a meal. And we speculate that nitrogen, since it's limiting in these environments, because there's not a lot that gets in there, nitrogen can be something that's allocated to the immune system of the mosquito. And there are some pathways that actually are in the mosquito already that are related to immune function that can be regulated through the addition of nitrogen. So we think what may be happening is that these mosquitoes that have lots of nitrogen as a larva, they're well fed. When they turn into adult and then they're given a viral blood meal, their body can fight that virus off and kill it before it actually replicates within their body. And we've looked at that both in the field and in the lab, and it seems to be consistent. Now, unfortunately, although all the mosquitoes that we sampled in Puerto Rico over that year and a half after Hurricane Maria, we set the heads aside and we did viral analysis on all of them, not a single one was positive for dengue. We were actually expecting to see a dengue outbreak after the hurricane for a number of different reasons. One is because it had already been about 10 years, which that's the cycle in Puerto Rico. It's about every 10 years. But more importantly, because there were fewer people with air conditioning because of no power, people were leaving their windows open and things like that. We were going to get more people exposed to mosquitoes with dengue, but we didn't see any dengue. So we couldn't really test it there in that circumstance.
So that leads into another question
Okay.
that came up is, why do you think there was no increase in adults following Maria? I mean, I would think there would be.
Yeah, so we did too. And all the anecdotal information is that there was. It's just that the hurricane happened in September. I applied for funding in October and got my check in January. So that's the quickest I could get down there to sample. And everything that I was hearing, it was the weeks or so after the hurricane when people were just being destroyed by mosquitoes. So, there's a couple things. One is that those habitats may have been short-lived. So you got one big burst of adults, but then the habitats start to dry up because there's lots of impoundment of water after the hurricane. Those start to dry up or get emptied. And that may have then decreased the availability of egg-laying sites for those females. And so even though there was lots of females, they didn't have a lot of places to lay. The other option is that homeowners were being more proactive about dumping material and also spraying, perhaps, to kill larvae and adults. And then the other option is just that, within that 3 months, the environment had changed sufficiently enough, the conditions had changed sufficiently enough to no longer support those high abundances. So we're pretty confident that there were within those 3 months. We just couldn't collect data during that time. We physically could not get to the island during that time.
Thanks. So this leads into my final question from Suzanne, which is what kind of educational resources are generally distributed to the public to help them understand and manage these to the extent that they can?
Yeah, so in terms of just mosquitoes broadly, at least in Puerto Rico, which I can speak to more specifically, there supposedly are various campaigns out there to educate the public. However, until about 4 years ago, there wasn't really a strong mosquito control program. Prior to about 2016, 2017, the mosquito control was handled through the health department, which was very incapable of actually doing systemic vector control. What you have to realize is that there are 78 municipalities in Puerto Rico. There are 104 small islands off the main island, many of which are inhabited, maybe 6 or 8 are inhabited. And so it's a very daunting problem, even though the island itself is only about 100 miles long and 30 miles wide. There is so much bureaucracy because it's like you have 78 states, and each state has its own laws and regulations, right? The other thing is that there's a lot of, in some cases, well-founded skepticism of any kind of government entities in Puerto Rico, and that has really limited the ability to have the government come in as an entity and say, we're going to do this across the board. So often it's left to the local municipalities to educate their population. That's changing a bit now. We actually tried to assist in that. We worked with about 70 different homeowners through that year and a half of sampling. Sometimes we couldn't sample at the same home that we sampled at last month or 2 months ago, so we had to find a new homeowner. So even though we had 48 consistent trapping locations, they weren't always the same place. And what we did is we worked with those homeowners. We gave them pamphlets that would educate them on what we were trying to do. And then at the end of that, we also showed them what the data were. Now, we didn't tell them about individual homeowners because that would, violate IRB approval and stuff like that. But what we said is like, this is what your neighborhood looks like. This is how many mosquitoes your neighborhood has compared to some other habitats. And these are the kinds of things that you can do to help limit those individuals and reduce those numbers. And many of them already have good education in terms of this. It's just the implementation is often difficult, especially in these low economic neighborhoods where they don't have control over, for instance, when the trash gets picked up or how much of it gets dumped in those neighborhoods. I mean, you drive around, there's tires everywhere because people are dumping them there, right? And there's not that really strong infrastructure to keep that from occurring. There's a massive problem on the island right now because of COVID and because of some other things. They haven't been able to get tires off the island. When I was there about 3 weeks ago, we were driving by piles that probably had 1,000 tires in them because they can't get them off the island. They're just being piled and piled and piled. And those are all habitats for Aedes aegypti, unfortunately.
All right. Well, everybody, let's thank Don again so much for taking the time to come and share his cool research with us.
Yeah, I really appreciate the invitation and chance to talk to everybody about ecology, which is, that's cool.
We think so.
Yeah.
All right. Thank you.
Yep.